is Bianca Dang, and today I'll be presenting about a non-invasive cognitive neural prosthetic that may improve amygdalar cerebral perfusion. I work at UCLA's Department of Psychiatry under my mentors Dr. Taylor Kuhn and Dr. Susan Buchheimer. Neurostimulation is an emerging therapy in treating psychiatric and neurologic disorders such as DBS, or deep brain stimulation, and TMS, or transcranial magnetic stimulation. However, most non-invasive transcranial stimulation techniques are limited by the range of disorders they can treat due to their inability to reach deeper regions in the brain. Currently, it's only achievable to reach these deeper regions in the brain via non-invasive, high-risk, and neurosurgical intensive deep brain stimulation methods. While highly effective, DBS requires neurosurgical interventions in order to occur, and for this reason, it is difficult because of the financial and physical requirements it has. TMS, on the other hand, does not have as much risk as DBS because it doesn't require neurosurgical interventions. However, TMS is also unpredictable in terms of its efficacy. So ideally, we would have a neuromodulation treatment that can reach deeper brain regions with the minimal risk component, like that of TMS, but with the high efficacy of DBS. That's what I'm here to present about today. Transcranial Focused Ultrasound Sonication, or tFUS, is targeted, effective, and non-invasive. In fact, it's the holy grail of brain stimulation treatments because it's a tool that's cheap, portable, and painless, and it can focally modulate deeper regions in the brain. In fact, Mark George has even gone to say that it's the next big thing. tFUS operates via ultrasound, which are acoustic energy waves with frequencies higher than the human ear can hear, and it can penetrate the skull and reach and exert effect on deep neural tissue with a high spatial resolution. tFUS, like other neuromodulation techniques, can be applied to psychiatric and neurologic patients and provides advantages over pharmacologic treatments because of its focal and direct impact on neural circuits. Furthermore, tFUS appears to be safe for neuromodulation in humans and is generally well tolerated. Unlike high-intensity ultrasound, tFUS acts on a non-thermal mechanism and does not have any negative effects on histology, the blood-brain barrier, or behavior. Early histology and animal studies of tFUS have demonstrated its ability to produce reversible physiologic effects on neuron clusters, including increased activity in targeted areas. In fact, fiber tracts and neuronal somas are more sensitive to the mechanical effects of tFUS, acting largely on voltage-gated ion channels. Using tFUS, neuromodulation in vitro hippocampal neurons via induction of voltage-gated calcium and sodium channels led to increased spiking activity and have been successfully demonstrated. Using real-time fMRI of rabbits, tFUS has produced increased activation in bold levels signals in regional brain targets. In monkeys, researchers were able to find a disruption of amygdalar function, both in resting state functional connectivity and amygdala-mediated behavioral tasks. To study this study sought to determine whether MRI-guided low-intensity tFUS can selectively increase regional cerebral perfusion in the amygdala. We hypothesized that low-intensity tFUS treatment will result in an increased blood perfusion in targeted areas and its functionally connected regions. So, to study this, we designed a double-blind crossover study that was randomized with within-subject controls. We had 6 participants who were half female and half male, and we had them come in exactly 2 weeks apart for tFUS sessions, one session targeting the left entorhinal cortex and the other targeting the right amygdala. Before each tFUS treatment and after it, we conducted a series of neuropsychological and psychophysiological assessments. We used a transducer that was circular, single element, and spherically focused, with an active aperture of 61 millimeters and 65 millimeters in nominal focal length, with a hydrophone measured -6 decibel focal width of 4 millimeters in water and -6 decibel with depth range in 50.4 to 80.5 millimeters. The tFUS was a BX Pulsar 1002 by Brain Sonics Corporation. The parameters for it was 5% duty cycle always, with tFUS targeting the amygdala at 10 Hz PRF for inhibitory and at 100 Hz PRF for excitatory for the entorhinal cortex. And on the left here, you see the shape of the tFUS wave focus. tFUS treatments were about 90 minutes long in the scanner, which was a Siemens 3T Prisma MRI. We used a targeting protocol by Spivak and Kuhn, and we used this to target the right amygdala on the top in the figure on the right, and on the bottom, the left entorhinal cortex. And by targeting these two regions, subjects served as their own subcortical controls. You'll notice a white dot on the outside of the skulls in both of these figures, and that is the tFUS device. We collected data by acquiring ASL images, or arterial spin labeling images, before and after tFUS treatment. The resultant sequences were processed using FSL and then subtracted to create subtraction images, and then overlaid on a high-resolution structural T1 weighted image. What we found was that ASL shows selective regional perfusion at tFUS-targeted regions and its regions functionally connected areas. And what's particularly exciting is that we see this not only in just some of the subjects that we treated, but all 6. And what's more, the ASL results did not show perfusion in the corresponding control regions. That is to say, when we targeted the amygdala, as you can see in our figure here, the amygdala-focused tFUS treatments increased perfusion in the right amygdala, and only the right amygdala, and not in the entorhinal cortex. When we targeted the left entorhinal cortex with tFUS, only the left entorhinal cortex increased in perfusion, and not the right amygdala. As such, this suggests that ASL can readily detect increased perfusion due to tFUS insonicated targets with a well-defined and discernible vasculature. This was also seen at the group level and effect size analyses that we conducted, and this demonstrates that tFUS has a large effect size on regional perfusion. Why is this important? The amygdala receives its primary source of arterial blood through the anterior choroidal artery. As a result, increases in ASL perfusion of the amygdala will cause an increase in the amount of blood flowing through the arterial choroidal artery. This results in a strong and readily detectable signal in the ASL MRI, and that is only localized to the arterial choroidal artery. Similarly, the entorhinal cortex is provided its main supply of oxygenated blood through one of the posterior choroidal arteries. Looking forward, little work has been done on tFUS neuromodulation in humans, and for this reason, it's potentially exciting that tFUS could become a neuromodulation treatment for the future. Its potential for treating symptoms of anxiety by modulating the amygdala are very exciting, potentially, and in mitigating Alzheimer's disease by directly enhancing working memory with the entorhinal cortex. Furthermore, the safety, accessibility, and non-invasiveness of ultrasound are incredibly and potentially promising for a vast variety of patient populations. Of course, our sample size was very small, and so replication of this study is necessary. Going forward, the relationship between the increase of perfusion and tFUS-related changes in functional connectivity and behavioral outcomes remains an important focus of our ongoing investigations, as well of course, in expanding this preliminary research due to its small sample size. But overall, we are hopeful and we are very excited. Here are my references, and I would like to thank my mentors, Dr. Susan Bookheimer and Dr. Taylor Kuhn, for making this possible, as well as our lab and our collaborators. Thank you so much, and have a wonderful day.